So our next speakers, we actually have 4 speakers that are coming up, but they promise to stay within their allotted time, I swear, is Dr. Albert Keung and Amay Bandekar. Dr. Keung is coming to us as an associate professor of chemical and biochemical engineering at North Carolina State University. He applies synthetic biology approaches to advance knowledge and treatment of neuroepigenic disorders. His work includes engineering new models for Angelman syndrome research and therapeutic testing, using human stem cell models to study these neuroepigenic mechanisms. And Dr. Bandekar is working with Dr. Keung, and really Dr. Keung brought him to us in order to really advance our preclinical models in order to be able to test drugs for Angelman syndrome in a more high throughput way. So he's an assistant professor in the Department of Electrical and Computer Engineering at NC State. How does a computer engineer get involved in genetic disorders? Well, he really is trying to understand and work through the interface of electronics, material science, and biology. So he's a really smart guy. To realize the next generation of sensors and energy devices in order to be able to look at organoids and how you can look at their changes over time with drug testing. So he's really working on this in Angelman syndrome lines, and this is a very exciting time for them to share their work, and they're bringing their students with them in order to really highlight the incredible work that these young trainees are doing and how hard they work and how much they care. So we thought it was really important that all of the funding we give them really goes to helping their students learn and do this type of work and be the next generation of Angelman syndrome researchers. So we wanted to introduce you to all of the fruit of their labor. So thank you.
Okay, thank you, Allison, and thank you to FAST for organizing the summit and all of the incredible hard work really guiding and ushering the translational research infrastructure and community. Let's see here. I think, okay, there we go. So we really had 2 goals today. So one goal is to describe, hopefully clearly, what the work we're doing in the lab is and how we hope it will impact your kids. And the second goal, which I'll start with, is really to, I guess, share a feeling with you all that Amay and I think all the investigators here share, which is that we have a feeling of a mixture of encouragement, hope, and faith in progress for the science that we're doing. Largely because, for one primary reason, it's the bright scientists that are in the lab doing the work, and they offer a lot of energy, motivation, optimism, and passion. And so that's one of the primary reasons we wanted to have them actually tell you a little bit about what they're doing. They'll each do a couple slides. And in that way, hopefully share that same feeling that a lot of the investigators in this room have and derive from them. Just really quickly, this is Begum and Chris, 2 seats down, and they're both chemical and biomolecular engineers, but they also they're mixing bioengineering wet lab expertise with bioinformatics. And Navya Mishra right there is a material scientist and electrical engineer, and now a synthetic biologist as well. And Tyler Johnson, next to Amay, is a chemical engineer. Actually, he was a biochemist by training, but has come and seen the light in synthetic biology and bioengineering, and actually is a PhD student in chemical engineering now. So let's see, is this advancing? Can we advance one slide? Oh, there we go. Okay. So, I mentioned their backgrounds because to tackle complex challenges, it really takes a diverse skill set of expertise. And so that's why I kind of mentioned their backgrounds. And these are just a couple pictures of our teams at NC State many of them are not here today, but four of our students are here to represent. Okay, so I am going to kind of set things up for them. So our research groups really are tackling three needs for experimental models. So one is to reflect the diversity of Angelman syndrome. So Allison already has talked a lot about this, but yeah, we really need to be able to address whether different forms of Angelman will react to different types of therapeutics, and we need models to be able to test that. The second is motivated, we need human models, and this is really motivated by a number of things, but I'll just point out one, which is that both the National Institutes of Health, the NIH, and the Food and Drug Administration, the FDA, are pushing more and more towards human cell-based models for preclinical testing. And so we want to make sure that we're maintaining and keeping up with that curve, so that it doesn't slow us down in the preclinical phases. And then lastly, anything that we can do to increase research speed and throughput, we're hoping to advance that. So I'll start off with this first need of reflecting the diversity of Angelman syndrome. So what the approach, there's actually a couple of approaches to do this, and I'll mention Dr. Yonghui Zhang at the end because he's been very helpful in providing cell lines. And they're taking one approach. This approach that we're taking is where you can start with one human stem cell, okay, right down here in white, right? And you can take molecular tools to snip in and out different pieces of DNA into those cells. And then you can create these other colored cells. And what's important about this approach is that these changes that we're doing to the genome, these molecular SNPs and edits, they're meant to mimic different forms of Angelman. However, everything else about these cells is the same. So this is what we call isogenic, meaning same genes. So other than those Angelman-related changes, everything else is the same about these cells. And why is that important? This helps reduce variability between experiments. And so if you want to compare one genotype, one form of Angelman to another, or a control versus a treatment, this can help reduce that level of variability. And it can facilitate, therefore, comparisons between researchers in institutions and companies. We can use cell lines that are not restricted by federal funding rules. So we can leverage NIH and NSF and use FAST funds as a multiplier effect. And then we're doing this in a way that will be customizable and shareable and fast in how these models can be engineered. So Begum's going to kick us off with one of these forms of Angelman, large deletions, and how she's taking an approach to tackle that.
Thank you, Dr. Keung. As we all know, UB3A is critical for Angelman syndrome. However, in class 1 and class 2 large deletions, not just UB3A, but many genes surrounding it are lost. And we want to study all of these genes. And because targeting these genes might be helpful to address some of the symptoms of Angelman syndrome. Hence, in our lab, we are trying to create a platform to study all of these genes. And as the basis of this platform, we are using cell lines obtained from Angelman syndrome individuals provided by our Angelman syndrome community. We are trying to put back the genes lost into these cells and study what is changing. But we are trying to do this in such a way that this platform is customizable and can be easily used by our Angelman syndrome community. Hence, before adding these genes back, we're trying to add something called landing pad into the cell lines. So let's go back to the meaning of the landing pad. So landing pad is a region where a helicopter can land on or take off, right? So same logic, but this time the helicopter is the gene that you want to study. So once this landing pad is integrated into the cell line, this provides a location for the gene that we want to study to land on. and it facilitates the integration of that gene. And the genes that we want to study are the genes lost in Angelman syndrome individuals. In addition to large deletions, we also need models for UPD and ICD, and now Ty will talk about the model that he's working on.
Thank you, Begum. Up to this point, we've mainly talked about the more prevalent forms of Angelman syndrome, namely the deletion class, but there are also less prevalent forms of Angelman syndrome, such as UPD and ICD, that are just as important to study and understand. So throughout this weekend, you will hear that neurotypical individuals have a single copy of UBE3A, while on the maternal chromosome of 15 in neurons. But in cases of UPD and ICD, both copies of UBE3A are present but suppressed by disruption in the regulatory or the imprinting. So our goal in this project is to use landing pack technology that Begum mentioned, as well as CRISPR-mediated epigenome editing to create cells that mimic the UPD and ICD Angelman syndrome children and their epigenotypes. Our hopes with this project is that we can further understand how these disruptions affect and contribute to Angelman syndrome, as well as potential therapeutic strategies. And now up to this point, we've talked about models that we are currently developing, and I'd like to invite Chris to talk about how we can use them to better understand human neurons.
All right, thanks, Ty. And so, so we're working with human cerebral organoids. And so these human cerebral organoids are these 3D microtissues that are used to model human neurodevelopmental diseases and neurodevelopmental disorders, including Angelman syndrome. And so as you can see here, the order of these cerebral organoids is a couple millimeters in diameter. And so these organoids are generated from stem cells, and one of the big advantages of these organoids is they contain many different types of cells, both neuronal and non-neuronal. And so in our lab, we're interested in looking at the cell type specific effects of UB3A absence and the absence of the genes around it. And so what we're doing is we're generating organoids from different cell line backgrounds, including neurotypical, some isogenic genetically modified cell lines, and even a couple Angelman syndrome patient-derived cell lines. And what we're doing is we're applying this technique called single-cell RNA sequencing, which allows us to look at which genes are being expressed in individual cells. And by looking at this gene expression, we can actually determine what type of cell we're looking at. And when we compare this gene expression profile to our neurotypical samples, we can actually see which signaling pathways and cellular processes are being affected. And all this with the goal of looking at how different cell types are being affected by UB3A absence and the genes around it. And so with that, these cerebral organoids are going to provide powerful tools in Angelman syndrome translational research, but we also need methods that are high throughput in nature to test these models. And so what I mean by high throughput is we need to be able to test a lot of samples, a lot of drugs, and a lot of therapeutics simultaneously. And so now Dr. Bandekar will come talk to you about one of these high throughput methods.
So, so one of the initial phases of drug discovery usually has like three main key pieces. Like the first one is synthesizing a bunch of potential drugs that could be useful for treating a particular condition. The second piece is something that Dr. Keung and his students just talked about, is developing some sort of like a model, like the brain organoid model on which these new drugs can be tested. And the third piece is developing testing platforms that can be used to understand how these new drugs interact with these models and whether they have the kind of efficacy that is required. So I'll be briefly talking about the third piece, the testing part of it. As Alison pointed out in one of her slides, we start with like 15,000 potential drug candidates, and then eventually, just a few hundred of them or so eventually end up in the preclinical and the clinical phase. So a lot of testing is required, and so you need platforms that can screen tens and hundreds of drugs on a really quick basis. And you have to do it in a manner so that you don't accidentally harm the brain organoids or any other models that you are trying to study. So here I show some of the examples that are either available in the industry or being developed in other academic settings. And as you can see, there are platforms that are either rigid in a fashion because that they end up damaging the model that you want to study, or they don't damage the models, but they are very difficult to fabricate and hence cannot be easily available to various other labs that are interested in testing these kind of testing drugs. So Dr. Keung and my lab, with the support of FAST, what we are trying to develop is a scalable platform that can be easily integrated with existing technologies that are available in various academic and industrial labs for fast screening of the various drugs. And with this, I'll hand it to our student, Navya, who is the person who actually does the work.
Thank you, Dr. Bandodkar. Okay. So on your screen here, we have the schematic of the scalable, high throughput, multi-organoid screening platform that we are working on currently. We have developed these thin and very flexible sensors that can be suspended in these wells of the 96 well plate that you see there, in which the organoids are grown. And in this way, these sensors can measure electrical and chemical signals coming out of these organoids. Now, why is it important? It's important because this will help us understand how these organoids are responding to the drugs being administered to them. If you look at the bottom left of the slide, you can see a very close-up, real-life picture of these sensors. Those black, hair-like things are the sensors, and indeed, they are as thin and flexible as a human hair strand. Right next to it, you can see a picture of the strands being incorporated with the organoids. So, as compared to the gold standards of sensing in neuroscience, we found that our probes are around 100 times more sensitive than the most commonly used glassy carbon electrodes and carbon fiber electrodes, as shown on the plot on the left. So, these are ultra-flexible, ultra-sensitive. They do not mechanically damage the organoids, and they can measure electrical and chemical signals coming out of the organoids. And most importantly, we can do this for 96 organoids at the same time, simultaneously and continuously over a large period of time. And we can do this without much complexity of fabrication or use. So our hope is that this platform will help speed up study and drug screening efforts towards Angelman syndrome. And here I have with me a 6-well prototype of this system, and if anybody is interested, I would be more than happy to share it with you. Thank you. Back to Dr. Keung.
All right, thanks, Navya. And just wanted to, I know you won't be able to read all of this, but there's a few other projects going on in the group related to Angelman, and including, we're starting to move some of these cell lines into partnerships and collaborations with other investigators and companies to do some testing of therapeutics. So just to summarize, these are kind of the 3 target features of the experimental models that we're hoping to accelerate Angelman syndrome with. And I just want to end with a few thank yous to well, in particular, I want to point out Yonghui Zhang and Stormy Chamberlain, who have provided iPSCs and stem cell lines that we're basing a lot of these edited lines off of. And I also want to thank FAST, of course, for funding, but also point out that the actual funds that FAST has raised and is supporting the research has an outsized effect beyond those funds themselves. These projects have been very exciting and motivating to many young scientists, and a lot of them want to work on this. So the FAST funds have actually recruited students that have external fellowships, they've been bringing in other funding, and so I just wanted to let you know that FAST is acting as a multiplier effect in many ways. And with that, I hope you see and experience the same hope and encouragement and excitement for the future that these young scientists are providing, me and Ame, and all the other investigators that run academic and translational labs in industry. So with that, thank you. Be happy to take questions.
Well, you guys are amazing, and it's really great to see such young minds so excited. That probably to all of us is kind of like science fiction, but the fact that you're able to figure out the drugs that make sense to us to then be taken forward into animals and into humans is what this is all about, and it all starts with models like what you're creating. So for that, we are so grateful, because this is just brilliant work that you guys are doing, and I hope you're proud of yourselves as well.
I had a quick question. Here. I may have missed it, but on the UPD, ICD landing pads, are you guys testing the overexpression of those other paternal expressing drugs that are typically not expressing on the maternal side?
Yeah, it's a great question. So we are we're establishing the state on the bottom where both UBE3A copies are silenced. And one of the concerns is, how do ASOs or even gene therapies or CRISPR-based therapeutics potentially interact with that? Would it turn on both copies of UBE3A and maybe lead to double the amount of UBE3A than you would have in a large deletion model? And so actually Ty is establishing the cell line, but the downstream work with the cell line is to throw ASOs on there, put indotecan, basically test a bunch of different potential therapeutics that have been investigated, and do a dose response. So we actually want to see whether it's controllable. If we add less or more of a drug, can we tune the amount of UBE3A that we're getting so that we don't overactivate or underactivate? We're not sure if it's possible in a UPD or ICD model, and we want to understand how they respond quantitatively.
That is awesome, and I love that de-risking the ASOs for paternal activation for UPD and ICD. But also, at least with UPD, there are some imprinted genes that typically don't express on the maternal side, that they're getting double expression, if that makes sense. So just like you're looking at the other genes and a large deletion, are you also looking at the overexpression of those imprinted genes in UPD?
Yeah, it's a great question. So on the bottom middle, that's Dr. Maria Fadri, and she is working on snow RNAs, which is exactly what you're talking about. So she's working on a system that you can titrate the levels of those, meaning you can tune it and control it and see what the effect is. So yeah, we're going after that.